Katja, you've developed some of the interactive screen experiments that the students might use. You call them ISEs for short.
Yes, we do. And the ones I developed are a digital fluorescent microscope and another ISE that it used together with it to analyze the images that are captured by fluorescence microscopy. Generally, microscopy is such a widely used technique in biology and also in health sciences. So that's why we continue to develop microscopy in the ISEs. There are various different types of microscopes, and they all do some different specialized jobs in the labs. And the fluorescent microscope allows to very specifically label certain proteins, certain structures inside cells in a much more detailed level than what students were able to do when they were using the digital light microscope.
So here's the microscope. What exactly is going on here?
So this one is a fluorescence microscope, and actually it's the fluorescent microscope we used to take a lot of the images that went into the digital fluorescent microscope. Like all microscopes, we've got a light source. The light is going through one of these cables here into the microscope. It's going through various lenses and objectives onto the sample that is here. If I want to see the cells in the sample, I can either use the eyepieces and look at the cells, but most of the time we're actually using a digital camera that is here on the side that is feeding the images to a computer, and we can look at the images on a computer screen.
You've got 4 fantastic coloured images over there. Can you talk me through them?
Yes, so here you see the images we're taking with microscopes like those, and what we're actually doing is we take all the different colors separately. So we first take this image, then we go to the next channel, take the next image, go to the next channel, take the next image. And then we're using software to create these multicolored images by overlaying all the 3 individual images and get this multicolored image.
So how does all of that translate into the digital microscope?
So as you might have spotted, the cells you see in the digital microscope and the cells you see on the screen here are pretty much identical. They're coming from the same sample, and these are the cells I used for the digital microscope. So here you see an image of all the colors and all the different channels merged. But in the digital microscope, you can look at each color individually, just like I showed you with the fluorescent microscope. So here you see the cell's genetic material in blue. Then you can see the cell's energy factories, the mitochondria, in green. And you can see the rest of the cell, the cytoplasm and the nucleus, in purple. And again, you've got the ability to overlay all of these channels. And just like in a microscope, you can change the objectives, but you can also change the zoom factor using a mouse and go in at all the in-between levels. So what we've got here is also the ability to show you some images of healthy cells and cells from patients with a disease, and students can investigate what structure of the cells has changed when a disease developed.
Using this digital microscope then, what kind of data do the students collect and how do they use it?
So what we're asking the students to do is quite often to go into these slides and to identify a particular structure inside the cells, and then to take an image that is illustrating the structure and put that in as into their reports as a slide. We do also ask the students to count and quantify the number of structures in certain cells. So on this slide, a very simple question could be, how many nuclei can you see?
So I count the nuclei, 1, 2, 3, 4, 5, 6, 7, 8-ish.
Yeah, or something slightly more complex. We hope that the students also interact with the wealth of other information that we've got. So in here, we've got a large number of other slides that we hope the students are going to interact with.
Now, what's really special about this is that the students get access to very rare and privileged material that you wouldn't just have in a normal lab.
Yeah, so for example, these cells are patient cells that are coming from a clinical study, so it's something that students would normally not have access to. We wouldn't be able to send out fluorescent microscopes to students in home experiment kits, but using these digital microscopes, students can have access to the same equipment that we are using in the lab.
Okay, sneaky preview. What have you got coming up?
Plenty. We continue developing these ISEs and digital microscopes a lot. So there are always new images in the digital microscopes. I've also developed a digital electron microscope, so students will be able to interact with electron microscope images in more detail. And that's coming up in a new cell biology module, but also will be involved in other modules. We've developed 3D models of cells, of plant cells, animal cells, bacteria that are going to be used in the new modules. And very excitingly, we are developing the first actual remote experiment for use in health and biology curriculums, where the students can decide on the treatment of a cell, and then we are measuring cell proliferation, and the students can come in and measure the rate of cell proliferation live while the experiment is going on here on the campus. And that will be the first remote experiment running in both the health sciences and biology curriculum.
So having all these ISEs means that students get a fantastic breadth of experimental experience, and you can do it all in your own time with complete access. That's great.
Yeah, it is.